Good afternoon, everyone. In our last class, we have been discussing about this optical microscopy. We have started our lecture on optical microscopy, and we have discussed about the different types of microscopic microscopes. First, we discussed about the bright field microscope. We had also discussed about the briefly about this polarizing microscope. We have discussed about dark field microscopy. Okay, and we have talked about several other types of microscopes. Okay, however, we have mainly discussed about the bright field microscopes, and from there we have seen what are the different components of this bright field microscope. We have also discussed the types of the bright field microscope. We have discussed about monocular microscope, and we have also okay. Then we have also discussed about the numerical aperture NA, and we have also discussed about the resolving power of an objective. Of an objective lens or a microscope. So these were the things which we have discussed in our last class. Now, I told you that there is one information, depth of field. Yeah, over here, the depth of field. So what is this depth of field? Okay, what happens over here? If you see, so the objective lens with higher magnifications, they normally have a higher numerical aperture and a shorter depth of field. So what do you mean by this thing? Now, suppose when we talk about this transmission microscopy, we usually talk about that we have to make our samples thin. Okay. However, how much thin you make your sample, they will be having a thickness. Okay. Now, as we are mostly interested in imaging this thin section, and we are analyzing all the rays which are or capturing all the rays which are coming out of this thin section, and we are using those rays for the imaging purpose or for the visualization purpose. Now, herein there can be two instances. Okay, the first instance is that these rays, they are giving us information about the whole slice. Okay, now in this case we say that the depth of field is higher. So when you get information about the whole slice, the depth of field, it is high. However, when we are using objectives with higher magnifications, in that case, what we are doing, say for example, we are looking into a very, very thin layer within that thin section. Okay. Now, when we are looking into this very, very, very thin section, then our depth of field it decreases. Okay. Now, basically, what is this depth of field? Depth of field means the thickness of your sample at which you are able to visualize. Whether you are able to visualize the whole slice at one time, in that case, we would say that the depth of field is very high. However, in certain condition, if we look into the sample and we are only looking into a very, very, very thin section of the thin virtual section of the sample, that means our focal length is within the sample somewhere and it is giving us an information about a very, 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 very thin section, then we say that the depth of field is very low. Now both of them, they have their own advantages and disadvantages. Now herein, if you see, with respect to the objective lens, we have discussed about the depth of field. Okay. Now, when we talk about the magnification of the objective lens or the power of the objective lens, if you see, I have discussed that the 200x, there are certain objective lenses which are 4x also. So in general it is 5x to 100x, but you can also find some objectives of 4x objective lens. Now among these objective lens also there are two categories of objective lens, and these categories of objective lens, they are classified based on the working distance. Okay, they are based on the working distance. One type of the objective, they would be having high working distance, and the other kind of objectives lens, it would be having a lower working distance. Okay. Now herein, when we talk about objective lens with higher magnification, basically we are basically comparing the objective lenses of a high magnification and a low magnification. Okay, suppose you are having a same category of the objective lens. So when we are increasing the magnification level, the working distance it reduces. Okay, the working distance it reduces. So when it reduces, when your magnification is increasing. On the other hand, it means if you have a low magnification objective lens, it will be having high working distance. Now what is this working distance? Let's come over here. So if you see, you are having an objective lens over here. So say this is your slide, and over this slide you are having a very thin sample, and then you are having a cover slip which I am marking it as black. Okay. Now then you are having your objective lens which you are using it to visualize the thin sample which has been marked in red. Now the distance between this objective lens and the top of the this cover slip, it is regarded as the working distance. Okay. Yeah. Now, as I told you, based on the magnification, we have seen how the working distance it increases or decreases, and we have seen that higher magnifications values, since they're having low this working distance, they are having lower depth of field. Okay. Now suppose I give you two types of objective lenses, say objective lens A and objective lens B. Both are having a magnification of 5x. However, this 5x magnification of this the A lens, it is a high working distance lens, whereas the B is the low working distance lens. Now, which one would you prefer and why? Okay. Now, you need to understand that these both type of lenses, they have their own advantages and disadvantages. Okay. Suppose as a biologist, you have taken a sample of say pond water, and in pond water you will be having microbes. Okay. Now these microbes, they would not be stationary. They would be moving within this layer. Now this movement, this microbe, it is at the bottom of the what you call of your thin sample section. In the next stage, it may go up again, it may come down, it may go up and come down, and whatsoever, and it may follow a random path, and it can be anywhere. Now you want to keep a track of this microbe, in what way it is moving. So what you would like to do? The thing is that if in this case, if we use a large working distance objective lens, then it will be allowing you to capture the whole thickness of your sample. Okay. So, but the thing is that you will not be able to get, suppose if you want to find out what is there at a particular section of this sample slice, okay, that you will not be able to do it with the A objective lens. Now if you have to do this, then you have to use a low working distance objective lens whose depth of field would be lower, and it will give you an information from a specific layer. And when you would like to find out this kind of thing, say for example, taken a stem and you have section of the, from that whole section of the stem, you concentrate on a particular layer or particular focal point of the sample, or you want to look at a very thin section within that whole section what you have cut, and you want to look into that layer. So in that case, the lower working distance help you. Okay. So in the first case, when you are in the objective lens A with higher working distance, it will give you the information about the whole section of your sample, and it can help you if there are any mobile objects like microbes and they are moving across the thickness of the slide. It will help you to track it. Whereas in the second case, in the B type of objective lens where you are having a low working distance, in that kind of thing, it will be helpful for you if you want to look into a particular section within that thin section of the slice. Okay. So in this way, you will be able to find out what kind of objective lens would be better for you. Okay. Yeah, yesterday I have already talked about the dual gooseneck illuminator. Okay, and as I told you, if you are having a sample which is opaque in nature, and this one is opaque, it is not allowing any sample, any light to pass through. So even if you are having the source, you will find that it will not, these lights will not be able to pass through the sample. So this becomes an opaque sample. Okay. Now you need to see the topography or topograph. That means you want to look into the top of the sample, of the opaque sample. Then you can use this gooseneck illuminator. So what you will be having, you will be having this objective lens, then these dual neck goose illuminators. It may also have a single neck, single gooseneck illuminator also. So what they will do, it will illuminate the surface for you. So you will be able to illuminate the top surface, and from here the rays will be collected by the objectives. Now, when you want to use this kind of illuminators, you need to understand that your microscope is what do you call, it is attached with um, high working distance objective lens. Otherwise, you will find that you are not having, you are not able to illuminate the top surface properly. Okay. Do you have any questions till now? No. Okay. Now coming to the different types of microscopes and cameras, how we can image the system. Over here, if you see the first image that I am showing you, it is a trinocular microscope. Okay. Now you would say, sir, in the last class you have told that we have monocular microscope and a binocular microscope. Now what is a trinocular microscope? You need to understand, if you want to visualize the image that is formed by any microscope, the microscope would either be a monocular microscope or a binocular microscope. This trinocular port, it is basically used for imaging purpose. Okay, so this is dedicated for imaging purpose. Okay, now what is the importance of this? So if you see, if you, when you are doing your projects in your next semester, and if you are working on some microscopic techniques, then you would be interested in putting up some good quality micrographs. Now this triangular microscopes would be allowing you to take micrographs. Okay, the cost of this triangular microscope, it is higher than the binocular one, which is again higher than the monocular one. Now it would be your another question that, sir, if someone is not having a triangular microscope, Say for the example, if there is some crunch, financial crunch in the company, but if he does not have money to buy a particular microscope and the money to get a binocular or a monocular microscope, so will he or she be able to capture the micrograph? The answer is yes. Here, with digital eyepiece microscope camera, there are several digital eyepiece microscope camera which has dimensions which are similar to the eyepiece eyepiece of the binocular or the monocular. These are standard ones, eyepieces. So in general, it as eyepieces. Okay. Now these eyepiece camera, microscope camera, they can be fitted by removing one of these eyepieces. So over here, if you see, one of the eyepiece has been removed and that has been used to capture the micrograph. Similarly, in the monocular microscope, there is only one eyepiece lens. So that eyepiece lens, it can be removed and it can be replaced with this digital eyepiece microscope camera, and it will be able to give you some good quality images. Okay. However, in general, these digital eyepiece microscope cameras, they have nearly 40x magnification. Okay, when there is a 40x magnification, so you would say that if I use a 40x objective lens and then I use this digital eyepiece microscope camera, then my magnification would be again 40 times. So 600 times the magnification would happen. Okay. Now you need to understand that when we are doing this, when we are increasing this magnification over here, due to the some of the due to the what do you call limit of the glass optics, slowly your image will be starting to blur. Okay, because glass based optical system, they may not be able to handle this kind of magnification. Okay, so what you will be getting, the first one, you will be getting a blurred image, and the second thing is that another important thing is that you will be having a lower field of view. Lower field of view. Okay, usually what happens, as I told you in our last class, you may have an objective of 40x. However, in general, we use an objective lens of 10x or maximum of 20x, and in some cases it may be 25x. Okay, now when you are doing this, so what is the total magnification? It is only 400 times magnification. So if you see, if you compare 400 magnification, 400 times magnification and 600 times magnification, and if you look into the size of the camera sensor, suppose I bring down this what do you call, I bring down the magnification of this eyepiece camera somehow to say 10x or 20x to so that we can simultaneously cover the we can compare the results. So both of them, they are having the chips, they are having the same size. So in that case, in 400 case, 400 magnification case, what would happen? You will be able, suppose this is your whole sample area, you would be able to capture a larger field of view. You will be able to see what is happening to the microscopic structure on a larger area. However, whenever you are going for such high kind of magnifications, what you are basically doing is that you are restricting yourself to a very limited area, so you are not getting a generalized observation. Are you getting my point? You are not getting an information about the generalized stuff. So to avoid this, what can be done? To avoid this, we can have reduction lenses. These are the reduction lenses onto which, so the reduction lenses, they can be attached onto the microscope cameras, and by doing this we can bring down the magnification of the digital eyepiece cameras. Okay, so by doing this, depending upon what type of reduction lenses you are doing, suppose you are doing two times reduction lens, so that would be 40 divided by 2. That means you are bringing down the magnification of the digital camera to 20x. Okay, similarly, you have 1.5 times of reduction lens. So if you do that, it will be 40 divided by 1.5, whatever you will be getting. So you will be having that kind of stuff. So it would be around 30, I suppose. Yeah, 30x. Okay. So we will be having that kind of thing. Okay. So by employing these reduction lenses, we can bring down the magnification of the digital sensors. Usually in the current day we use the CMOS sensors, so we can bring down the magnification levels of the CMOS sensors. Okay. In the case of trinicular systems, this attachment that I have shown you, this is called C mount. Okay, this is called C mount. Okay, now this C mount, in these C mounts we can have these color CMOS cameras, microscope cameras, which are attached with the suitable reduction lenses. We can attach it over here. It would look something like this, and we will be able to capture the images properly without blocking our visual what do you call our visual observations through the eyepieces. When you are attaching these digital eyepiece microscope cameras onto the eyepieces, basically you are blocking one of the ports, at least one of the ports over here, and you can use this port for the visualization purpose. Okay, so when you are attaching or you are putting it into the this monocular microscope, again you are doing the same thing. Okay, however, if you are using the trinicular port, in that case what you are doing, basically you are not blocking these eyepiece lenses, and you are able to visualize the microstructures through your naked eyes. Okay, so if you have any questions, no, we can discuss. No? Okay. Now, till now, I have talked to you about the microscopes, where we have seen we have a source at the bottom, then we have the stage. Over that stage, we keep the sample, then we have the objectives, then we have the optical path or optical tube. Then we have the eyepiece. Okay. In these cases, we are able to microscope take the micrograph of most of the samples. However, if you want to say image mammalian cells, say in T95 flask, if you see what is happening over here, these flasks, they are having their own thickness. Now, if you have to put the objective lens, it would be very, very wide or higher working distance. You need an objective which is having a very high working distance. Okay. Further, usually we have seen when we are going for the this high magnification uh, microscopy, where we want especially which we need if you want to analyze the mammalian cells, okay, which are adhering onto the bottom of the T95 flask. In that case, we will not be able to use the higher magnification, okay, because in that case these objective lenses, they would be crashing into this what you call T95 flask. Okay, say for example, also if you have say multiplate system, multiplate culture, multiplate uh, a culture system, so where you are having 96 wells, so you will be having some wells like this. So in there will be a plate and there will be these things, wells would be there, and in within that you culture the mammalian cells. So over there also you will not be able to use the normal bright field microscope. In that case, what they have done is that the illumination system, this is the illumination system, has been taken up, and the objective system, if you see here we are having the turret, okay, that has been brought down, and then over the turret you will be having the objective lenses. Okay, so what it will do when you do this, it would allow you to achieve even very small working distance, and you will be able to visualize the mammalian cells properly. Okay. Now this kind of microscope where the optics have been flipped, what do you mean? That means the source has been taken to the top, and the objective system, the light collecting system, has been brought down. That kind of systems are regarded as inverted microscope. Okay, they are regarded as inverted microscope. So this type of microscope, they are basically used while you are doing any cell culture or tissue culture experiments. Okay, so this is one of the important thing. So this is also a bright field microscope. Please remember, this is also a bright field microscope, but it is an inverted bright field microscope. So if I ask you to write a short note on bright field microscope, bright field microscopy, you have to write about both upright means this original configuration system, this is upright bright field microscope, and the inverted microscope. Okay, so do you have any questions? No? Good. Further, in my last class, I have told you another important aspect. Suppose there is an opaque sample and you want to analyze the topology of the sample. So what would you do? In that case, I told you that there are microscopes which are regarded as reflection microscope. And I also told you that these reflection microscopes, they are also sometimes regarded as epi-illumination microscope. Epi-illumination microscope. So are you getting my point? In this case, so there is another type of microscope which is regarded as reflection microscope and epi-illumination microscope. Okay. Now, this is the architecture of this first one. This is the architecture of a reflection microscope. However, let us first see the path of light in the reflected light microscope. Then I will come back to this microscope. So over here, if you see, there is a light source. However, this light source, if you see, it is on the same side of the sample as that of the eyepiece. In the previous case, if you see, the source and the eyepiece were on the either side of the samples. Whereas in case of reflected light microscope or the epi-illumination microscope, the light source and the eyepieces, they are on the same side of the sample. Now there will be these optical systems which will be condensing the light and they will be processing the light, and these processed light, it will be incident onto the glass plate reflector. So this is a reflector and it is basically a 50-50 reflector. So 50% of the light, it would be transmitted through the objective lens onto the sample, and from this so this light will interact with the with the surface of the sample, and it will again be reflected back through this objective lens, and it will be passing towards the eyepiece. Okay, so this is the light path in the reflected light microscope. Now let's come to the architecture of this reflection microscope. If you see over here, at this end, at this fag end, we will be having the light. Okay, the light source, it may be the halogen light, it may be the LED source. Then within that we will be having the condenser. Okay, so condenser will condense the light, Then if you see, we are having various the filter holders. So these are filter holders. Okay. Then we are having aperture stop. So in the condenser, we have discussed about this apertures in our last class. Yeah. Over here there is a diaphragm and or filters. Okay, so this diaphragm, it is basically over here we use an aperture system. It is just like our pupil, so you can mechanically, so it will be a concentric system. So this smaller part, it is the blank part from where the light passes. Then there is a mechanical system from where you can increase or decrease the size of this aperture. Okay, then we can have some filter systems. So those filter systems can be added to manipulate the light, so that we can, manipulation means so that we can apply some band pass filters, optical filters, which will allow us to selectively select some colored light. So what are filters? uh, if you remember, suppose you are having a light source, and if you place a red color, this OHP shades, then over here you will be getting the red light. So what is this red light? You are basically this OHP shade, it is acting as a filter. Okay, and what is this red light? This is a band pass light. So you will be having only a particular band of light which will be passing through this filter. The same thing happens over here also. In over here you are having this filter slots. You can apply some filter, then you can have this aperture stop. So there would be a mechanism where you can increase or decrease the aperture. So through this you will be having a dichroic mirror. So this glass plate reflector, it is also regarded as dichroic mirror. Okay, so through this dichroic mirror, the light will be passed from here onto the sample surface. Suppose this is the sample, it will be striking over here, and again after striking, it will go back to the dichroic mirror. So then here you will be having a prism. Through this prism, the light will be reflected into your eyepieces. Okay, and this is how you can get it. Again, you can manage the focus. You are having this fine focus knob, coarse focus knob, and what you can do is that you can increase this height of these objectives. Basically what you are doing, you are increasing and decreasing the distance between the sample and the objectives. So in that way you will be able to move the mechanical stage up and down in this case, and also there will be an x direction and y direction movements, which is also regarded as mechanical stage control. Okay, in the case of reflection microscope, usually your stage moves up and down. However, uh, if you look into over here, here also you will be having these mechanical stages which will be moving up and down. However, there are certain bright field microscopes also where the objectives, it may be moved up and down. Okay. However, in the current day in technology, it is easier to move the mechanical stages up and down. Okay. So in this way, you can move it and you can focus it and you can visualize the thing. Again, for the imaging purpose, if you see, it is a binocular microscope. So you can move this eyepiece and you can attach the CMOS cameras for the imaging purpose. Okay. Then coming to the polarized light illumination. So in the polarized light illumination, if you see, suppose you are having two polarized shades and they are kept parallel to each other, so you will find the light it is passing through. Okay. So plane. So after this first one, you will be getting the light as the plane in between these two things. You will be having plane polarized light. Okay, and if you keep the first polarizing sheet and the second polarizing sheet parallel to each other, the light will pass through. However, if you put it in a put the second one 90-degree apart from the first stage, so plane polarized light will not be able to pass through the second polarizing sheet, and this second polarizing sheet in microscopy it is regarded as analyzer. Polarize. So the first sheet it is regarded as polarizer. So you need to understand polarizer, they would be near to the source, and analyzer they would be near to the eyepiece. Okay. Now over here in the bright field microscopy, what is happening over here, if you see the background of your samples, they are bright. Why this is so? Because suppose there are certain samples which you have kept and you are passing the light over here. So what is happening over here? So suppose there is a gap in between. So in between these gaps, the light will be passing unhindered. So these lights are unhindered. Unhindered light are being passed. Okay. So it will be having, when these light beams they are falling onto the sensor, they will be having an higher higher intensity levels. However, the light beams that are passing through the samples, what would happen? Their intensity would decrease, and the objects in the bright field illumination they appear as darker, whereas the background where there is no interaction of the light, there it is brighter in nature. However, when you talk about this plane polarized light, so what is happening over here? Let me draw this again. So you have the light source, then you have the polarizing sheet, okay, then you have the condenser. So I am not drawing the condenser. Okay, I am not drawing the condenser as it is now. Okay, so what is happening over here? When you are applying the polarizing sheet, only the plane polarized light they will be passing. Suppose at the top you have the analyzer. This is the analyzer, and at the initial stage this analyzer is having the same orientation as that of the polarizer. Then all the light will be passing through, and you will be getting bright field images. Okay. All the light means plain polarized light. Okay. However, in the second case, what would happen if you put it into the 90-degree cross polarized way? So what is happening? This light, which were initially passing through these gaps unhindered, they are not able to pass. Okay, and hence, if you see the unhindered or the blank spaces where there was no interaction of the light, there are no interaction of the light, they are being completely blocked. They are being completely blocked. However, if you see the samples or the objects herein, so what is happening? These lights, they are passing through the samples like this, and when they are passing through these samples, the orientation of these lights are changing. And when this orientation is changing, this cross polarized lights, they are this cross polarized analyzer, it is not able to stop the samples, and hence you are seeing that these are appearing as brighter. Okay, these objects, they are appearing as brighter. This is the this is how your polarizer it functions. Okay, so you can see why. I can give you a question, why your bright field image, it is having a brighter background, whereas in the cross polarized illumination you have the background as the dark as a dark you are having a dark background. So you need to explain all of this over there. Okay. Now there are certain simple polarizing kit which are available in the market, uh, using which you can upgrade your simple microscope, normal bright field microscope, to polarizing microscope. Okay, and we have also done it in our own lab. We have made our this system, we have made on our own, and we are using that thing with very good effect. Okay, so once you come back to the institute, we will be discussing about those. So this is the configuration of the polarized light microscopy. See, you are having the light, then you have the polarizer, then see you are having this plane polarized light which is coming, then you are having the birefringent specimen. Through this specimen when this plane polarized, through this specimen when your light, this plane polarized light is being moving, so you are having two types of rays, extraordinary rays which are maintaining the plane polarization, and the other one is the extraordinary ray where there has been a change in the polarization effect due to the refraction induced by your sample. And then you have the analyzer. So through this analyzer you are blocking the rays. Okay, so blocking this what you call plane polarized light, and you are basically getting the light which has interfered with the sample. Okay. So this is a system where you are having polarization both in the upright direction and also in the epi illumination system also. So in the epi illuminator you will be having a polarizer over here also. Okay, so this is this polarizer, okay, over here, and then you are having the analyzer over here. Okay, with this we will stop for today.